Hello, I'm Adrian and I am a student in Biology Agrosciences. I'm particularly interested in interactions between plants and pathogens and biological alternatives to limit the use of chemical inputs in crops. In this video, I'm going to tell you about biological alternatives for crop protection based on algae use. Since antiquity, many writings describe plant disease. Rust, a pathogenic fungus of cereals, causes lot of damages during antiquity around the city of Rome. During the medieval age and later during the rebirth age, many diseases are responsible of major famines. The burning pain caused by ergotism causes the deaths of millions of people who were consuming bread in Europe. Today, food scarcity is no longer the main problem and current uses are more economic. You probably know that plant disease can lead to significant losses of yield in any crop with sometimes dramatic economic consequences for farmers. Today the fight against pathogens is still very predominantly chemical. The main problem is that phytosanitary products accumulate in the environment and cause human health troubles and ecological problems like water and soil contamination, which leads to a soil fertility decrease. Now, to give you an example, the worldwide deaths and chronic diseases due to pesticide poisoning number is about 1 million per year. Furthermore, the excessive use of plant protection products is responsible for the development of resistances in pathogen populations. Today, the need to find biological alternatives for crop protection is becoming increasingly important. In addition to prophylaxis, management of the plant and its environment, or varietal selection methods, the use of biocontrol agents may be as effective as chemical treatment. Most fungicidal products are effective against pathogens, but they can also be phytotoxic for plants. In other words, they affect plant health, and this is a paradox because plants in bad health are more susceptible to be infected by pathogens. Biocontrol products are less toxic than chemicals and they are quickly degraded in the environment. They are effective in small doses and they are more specific to a biological target than pesticides. It's possible to use many natural ways to protect plants, like for example microorganisms like insects, microorganisms like fungi or bacteria, mediators to disturb insect reproduction, or natural substances extracted from plants. Algae are an interesting alternative because many species are not yet studied. Some substances from algae may have antifungal properties and other molecules may stimulate plant defenses. There is a large diversity of marine algae. They are brown or green that can be pluricellular like laminar algae or unicellular like cyanobacteria, and these all produce secondary metabolites potentially usable in agriculture. With this in mind, let's move on to an example of an efficient compound on plant protection: laminarin. Laminarin extracted from a brown alga, also called Laminaria digitata, is a plant defense elicitor. This molecule is recognized by a receptor of the plant as an attack signal and will induce defense reactions such as a strengthening of the cell wall as well as a greater production of phytoalexins. These phytoalexins, like for example resveratrol and viniferin in grapevine, can protect the plant against pathogen attack because they are able to inhibit pathogen growth. When laminarin is recognized by a plant receptor, there is also a greater production of PR proteins. These proteins are able to degrade the wall of fungi and bacteria. Other PR proteins have antifungal activities by interacting with the pathogen's plasma membrane. In fact, it has been shown that the recognition of laminarin by the plant triggers expression of 10 defense-related genes. This basically means that the plant is more stronger to fight against pathogen attack. There are also many other useful algae for plant protection. For example, Leysoional trabeculator, a brown alga, reduces the disease severity caused by Botrytis cyanera on tomato leaves. When extract is sprayed, it stimulates plant defense mechanism and inhibits tissues necrosis caused by the pathogen. This alga can also reduce the growth of gram-negative plant pathogenic bacteria. Another example is the Durvilla antarctica. Extracts obtained from this brown alga have antiviral effect by plant defenses stimulation. The tobacco leaves are protected against infection with the tobacco mosaic virus, and there is a decrease in the necrosis number on the leaves. What we find is that polysaccharides, main effective components in algae extracts, have antimicrobial and antifungal properties and could lead to cell lysis. Algae contain also large lipids quantity. These fatty acids can penetrate in the cell wall and kill microorganisms by leading to disruption of the cellular membrane. Fatty acids lead to membrane disintegration of bacteria, fungi, and yeasts. We'd like to finish by saying that algae provides many opportunities for crop protection and can contribute to an ecological transition in the future with using of more environmentally friendly methods. One final question this raises is to know how to implement these biological methods in large scale. Thanks for watching.